Transduction, it is a horizontal gene transfer technique. There are mainly three types of horizontal gene transfer technique in bacteria: transformation, transduction, and conjugation. Transformation that we have already studied. The second one is transduction. The speciality of transduction process, gene transfer process, is that it is mediated by bacterial viruses, also known as bacteriophages. There are mainly two types of transduction: generalized transduction and specialized transduction. Generalized transduction means here any portion of the host genome is packaged inside the mature virion. But in case of specialized transduction, only a specific region of host genome is integrated into the virus genome, and then it is packaged inside a mature virion. So here, along with the virus genome, a small portion of the host genome is integrated and it is incorporated into the mature virion head. That is the main difference between generalized and specialized transduction. Transduction is very common in some bacteria. For example, D-sulfovibrio, Escherichia, Pseudomonas, Rhodococcus, Rhodobacter, Salmonella, Staphylococcus, Xanthobacter, etc. Let us discuss in detail about generalized transduction. I told you that in case of generalized transduction, a portion of the host genome is as such packaged inside the viral head and it is transferred to the next bacterium. So, this generalized transduction, it is extensively studied by using two bacterium and phage. For example, if you are using Salmonella enterica, use the phage P22. Similarly, E. coli is another extensively studied bacterium for generalized transduction with the phage P1. Let us study the generalized transduction process step by step. This is the first step of generalized transduction process. Here you can see a bacterial cell which contains bacterial genome in green color. So, over the bacterial cell there is a phage attached. The attached phage then inject its genome into the bacterial cell. So, the bacteriophage genome here is in red color. You can see that the phage injecting its red colored genome into the bacterial cell. After some times of infection, you can see now the red colored phage genome is totally released into the host cell. Whenever the phage genome is completely released into the host cell, the phage genome replicates and produces more copies of their genome. You can see here more copies of red colored phage genome. At the same time, the phage genome also induces the fragmentation of host cell DNA. That is, you can see here that green colored DNA is fragmented. Initially, the green colored host DNA was in circular form. Later, the red colored phage DNA induces the fragmentation of green colored host DNA. By using the phage genome, that in previous image it is denoted in red color. So by using that phage genome, the phage synthesizes whatever proteins and RNA require for its multiplication. So the phage actively synthesizes the proteins required. After synthesizing all the essential elements from the phage genome, the phage, by using all those required or all those synthesized material, phage actively synthesizes their own progenies. You can see here, okay, so many progenies were produced. So this progeny contain a head. The head should be packaged with a copy of phage DNA. Okay, so here this progenies were packaged with a copy of phage DNA. But by chance, any one of the progeny or some of the progeny, their head may be packaged by mistake, packaged with the fragmented portion of the host genome. Whenever the mature progenies, viral progenies were synthesized, they were released out of the cell by cell lysis. So this type of bacteriophage life cycle is known as Lytic cycle. So in Lytic cycle, phage particles were released by the host cell lysis. You can see here. So two types of progenies were released. One type of progeny, they are containing phage genome in their head. Another type of progenies, they are by mistaken packed with the fraction of bacterial or host cell genome. So they are known as transducing particle. They contain the host cell or bacterial cell DNA. Later, the newly synthesized progeny, that is the transducing particle, especially the transducing particle, which containing the bacterial genome in their head. So they are known as transducing particle. So you can see here transducing particle. So whenever they released out of the cell, they infect a new bacterial cell. To that newly infected cell, bacterial cell, they inject their genome. So here the transducing particle genome is genome of the host DNA or bacterial DNA. So the previous bacterial DNA through this transducing particle now transferred into the new bacterial cell. You can see here a green colored genome is transferring by the phage. The released genome, that is, later undergo homologous recombination and it is integrated into the bacterial cell genome. So you can see here previous bacterial genome is integrated into a new bacterial cell by means of a bacterial phage or bacteriophage. Okay, so the new cell that contain two types of genome. One is its own genome, another is genome from another bacterium. So such type of cell is known as transduced cell, and the overall process is known as generalized transduction.